Let me see. Okay, it works. Okay, good morning, everyone. This is Ginger, the Managing Director from MSQ. I'm delighted today to have such a esteemed speaker on this panel to discuss very exciting field of protein degradation. So actually, we have a very good mix of participants. Today, we have big pharma, we have startup companies, so we have mid-cap public companies. And also each speaker have different roles, including CEOs, BDs. Some of them have a really, really strong science background. And also location-wise, we have people from United States, Europe, and China. So this is indeed a very global, international panel. So, so I believe each speaker today can contribute a highly unique perspective and insights into the global developments in this emerging field. And also we will heavily talk about partnership opportunities. For example, how to leverage partnership as a complementary to the in-house development. So without further ado, I would like to invite each speaker to briefly introduce themselves and also their companies. So I will go with the sequence on my screen. Tom, do you want to take the first position?
Sure. Thanks, Ginger. So I'm Tom Shepherd. I'm the CEO of Captor Therapeutics, which is a European public TPD company. I believe probably the only one. We're listed on the Warsaw Stock Exchange, which might be new to some people, but it's a very vibrant exchange for biotech. The last 7 years, we have around 110 FTEs in the company. I've been working in biotech for many years, both in the US and the Bay Area, also in Boston, but more recently I came back to Europe as I'm originally from the UK. Some of the Anglophones might recognize a Scottish accent. So I'm enjoying my life now in Poland with this company. And it's really interesting here because we have been able to put about $90 million to work in Captor. About half of which from investment, half of which from non-dilutive funding. So we really have built a capable platform with quite a talented team. I guess where we are is we have a molecular glue for hepatocellular carcinoma, which is quite a unique offering, which is IND enabling studies, and we're expecting to file a CTA by the end of the year in Europe. And I guess the thing about Captor is we have a platform which really works both with molecular glues and with PROTACs, as I've noticed some of the other companies do here today as well. But as well as that, we are really got a big focus, and I think it's a differentiator on structural aspects. I think, in our platform, we work obviously with ligases and target proteins and also small molecules. And I think we have solved about 400 crystal structures in the last couple of years. So we really do focus on structure-guided screening and optimization, which I think is a big differentiator of our approach. And of course, new ligases is a hot topic, and we'll come back to that later, but it's certainly something which is at the heart of what Captor is doing as well. And I'll maybe mention that later. So in the interest of time, I think that's probably enough for me, and I'll pass on to my next colleague.
Okay, thank you, Tom. I do like your accent. Okay, next one, Scott, you want to take over?
Sure. Thank you, Ginger and MSQ, for the opportunity to be here. My name is Scott Boyle. I'm been with C4 as Chief Business Officer about 18 months. C4 Therapeutics was founded in 2015 with a focus on doing targeted protein degradation in oncology. Similar to others on the panel, our strategy involved both non-dilutive capital through partnerships where we've had collaborations with Roche, Biogen, and Calico to discover molecules in therapeutic areas largely outside of oncology with collaborators, while we've advanced our own internal pipeline in oncology. We just recently announced that we've had our fourth IND approved for the US. So we have 3 molecules in the clinic with a fourth coming soon. one is a molecular degrader, and then the other three are bifunctional molecules. And so similar to Tom, a focus across there. I think one of the things that drew me to C4 was the ability that the scientists have been able to get orally bioavailable molecules of these large molecules. And so certainly excited to be here at this time. And then with, I guess, background, I'm a biochemist before moving to the business side, where I've been involved in strategy and BD for a number of years.
Thank you, Scott. Now, next, Xiaobao. For those who don't know the character of Chinese, like Xiaobao means little baby in Chinese. That's a very lovely name. Xiaobao, you want to introduce yourself?
Okay. Thanks, Ginger. Okay. Our company was registered in 2020 and operated officially in 2021. Since at the beginning of the foundation, we have determined our differentiation from many other companies, which is the GluTech platform, the dual platform combining the advantage and know-how of the glue and protect, which has also brought us more insight about how to design high selectivity ligands and improve bioavailability much more efficient. The differentiation strategy makes us emerge from the market and help us attract some investors. We have pipelines both glue and protect and have just become a clinical stage company. In terms of the process of finding IND, our rapid advancement benefits from the focus of the committee of the team. We have established the artificial intelligent virtual screening platform, in vitro pharmacodynamics screening platform, pharmacokinetics platform, proteomics platform, and in vivo drug pharmacology evaluation platform, and have constructed a complete whole process drug R&D system. We carefully managed our schedule and designed the preclinical study timeline in the most efficient way. Our CRO has also dedicated to refine on the project management of our pipeline. Our joint efforts make our first IND filing work smoothly. We welcome any potential collaboration both domestically and internationally.
Thank you, Xiaobao. Yeah, looks like you do have a lot of quite a few exciting platforms, and we can go back to this with more details later in our conversation. And Michael, you want to take next?
Sure, thanks, Ginger, and pleasure to be on this panel today. So Michael Paladinetti, I'm the Global Head of Business Development at Sanofi. My team focuses on early to late stage deal making in our core therapeutic areas of oncology, immunology, neurology, rare disease, vaccines, and also supports our general medicine business. We have a separate and dedicated group of professionals that also looks at innovative platform technologies, such as targeted protein degraders. So I'm very much active in the space. I'm based in Boston and have been with Sanofi for 6 years and previously held business development roles in large pharma at other companies such as Novartis, Takeda, and BMS. Great to be here. Thanks.
Thanks, Michael. Yeah, Scott is also in Boston. I mean, I visited the C4 office. That's really nice place. Yeah, Michael, if you have time, you should pay a visit. All right, Leo.
Thank you, Ginger. This is Liu Fu, the co-founder of Glue Therapeutics. Glue Therapeutics was founded in March 2021, which is almost 2.5 years ago. The company headquartered in San Diego, California, and we also have both research operation labs in both San Diego and Shanghai, China. So now Glue Bio has developed a robust TPD platform, which is designed and operated by members with years of experience and deep knowledge in developing TPD drugs. Just like Leo Morph or Tree9 Bioscience, we have a team of key members from BMS COG and also Johnson & Johnson. And so all of the key members has like 6 to 8 years experience in TPD area. So regarding to the pipeline, we have 6 assets in our pipeline. We go to the first IND approval by FDA in April this year, which is the Merck group drugs for treating liquid tumor and both liquid tumor and solid tumor. And we just filed the second IND filing last month by end of June. And we also have the third asset, which is the first-in-class program for multiple solid tumors and in every stage. We also have several assets and AO stage for different therapeutic areas. I guess all of them are Merck and well. So you can see Global is really focused on first-in-class. I think most of the assets are in first-in-class targets, and many focus on Merck group. So we have only one program. So yeah, thank you for inviting me, and very nice to meet everyone here, I guess. Yeah, thank you.
Thank you, Dio, and congratulations on the approval. Okay, thanks for everyone for the nice introduction. So next, what I'm going to do is I'm going to ask a few questions specific to each speaker before we move on to a group discussion. So I would like to start with Michael. So Sanofi actually, so as you just mentioned, right, Sanofi is one of the big pharma with heavy investment in the TBD sector. And you just mentioned you have transactions with Chimera and also have a partnership with Lyrics. So my question is that, can you shed some light on these transactions and how you see they align with Sanofi's overall strategy and your future plans in this field? And lastly, I believe the audience really, would love to know the criteria that make a deal quite attractive. So, so the last question is from a large pharma perspective. Perhaps you can also comment on that as well. Yeah, thank you, Michael.
Sure, thanks, Ginger, and happy to comment on these deals with Chimera and Nurix. So both of them were announced in 2020, and they were, they addressed critical gaps across multiple therapeutic areas in our early pipeline. I was directly involved in both of these business development efforts, and I'm very proud of the collaborations we've established with each of these companies. I'll start with the Nurix. So, in this case, we were interested to leverage the company's proprietary drug discovery platform, including its DNA encoded libraries, to create novel small molecules designed to induce degradation of specified targets. So we kicked off the collaboration with a selection of 3 targets. And a year later, we added 2 more. So the totality of the effort now is across 5 targets. So these are hard to drug targets that have been elusive to conventional modalities in the past that we think are particularly suitable for a PROTAC approach. Switching to Chimera, then, the story there is somewhat different in that relationship centers around the company's first-in-class IRAK4 degrader program, which is being developed for multiple immunology and inflammation indications. So in the case of Nurix, the discovery collaboration, in the case of Chimera, very much focused on established programs. For those who are unaware, IRAK4 is a key protein involved in inflammation mediated by the activation of toll-like receptors and TL1 receptors. The role of these pathways has been demonstrated in several inflammatory and autoimmune diseases, including priority indications like atopic dermatitis and hidradenitis suppurativa. So areas that Sanofi is very much interested in bringing new innovation to. In this case, the structure of the relationship was such that Chimera took the program into the clinic, and at the end of last year, released positive results from their phase 1 clinical studies. Based on these data, Sanofi now will be taking the lead and advancing the program into phase 2 clinical studies. So we're very much looking forward to the future story there behind the KT-474, the IRAK4 degrader. Looking ahead, beyond these 2 collaborations, like as you're saying, and we're committed to the space, and we're very much interested in exploring additional collaboration with leaders in the targeted protein degradation field that have proven differentiated platforms that could hopefully unlock new indication spaces like neuro, for example, and hopefully would generate the next wave of first-in-class, best-in-class medicines to address high unmet medical needs. And finally, to your question about what makes the deal attractive, well, the honest answer is everything. But I think a good place to start is a compelling differentiated science, which has the potential to generate, as I say, these next generation of first and best-in-class medicines, supported by the right people around the table that are committed to making a collaboration a success and unlocking value for both partners. So in the end, all the components of the deal need to come into place from an R&D, commercial, legal, finance, et cetera, perspectives. So it's unfortunately not one thing, but a multitude of factors that need to come together in the right way to compel us to move forward with a new relationship. I'll pause there and happy to expand later.
Thank you, Michael. Yeah, and it looks like you guys have really already have a fruitful collaboration with Chimera and New York. That's quite nice. And also, it's interesting you mentioned CNS as another direction for protein degradation. Actually, there's an upcoming TBD summit in Boston in September, I believe. Actually, they have a whole section designated to talk about the opportunity in CNS. That's probably quite interesting. Yeah, thanks, Michael. I'm sure your insights are quite inspiring, especially for those who are looking for partner opportunities with big pharma. Moving on, my next question is for my friend, Scott. First, congratulations on the landmark transaction with the Beta Pharmaceuticals. And also, I saw the news, you guys already have the IND approval for the EGFR. Yeah, so double congratulations here. So my question to you that can you talk about the early motivation for C4 to partner with a Chinese company? Because as far as I know, this actually is the very first one in the space of degradation for a TBD company, partner with a Chinese company. And also, what is your perceived potential of a Chinese market for 8919? And the next, I think it's gonna be very helpful if you could help, offer some guidance on those who are also looking for China partners. Finally, maybe you can also comment on the because you guys in the Beta is going to conduct a multi-regional clinical trials, right? So you can also comment, on the benefits and the risk associated with conduct multiple regional clinical trials in both China and the United States. Yeah, that's my question to you, Scott.
Yeah, for sure. And again, Ginger and I collaborated on this transaction, so certainly happy to give the perspective which started it off, right? So I think the main motivator for us to seek a partnership for CFT8919, which is our EGFR degrader in China, even though we were at the pre-IND stage, was driven by three main reasons. The first one is, there's just the opportunity for a company of our size to serve as broad a patient population as soon as possible and access those patients. Second is we think there's a chance to accelerate development, and I'll circle back to that. And then third, I think the other part is that a partnership at this stage allowed us to share risk and access expertise in a capital efficient market. One of the key insights on EGFR is there's a much higher prevalence among Asian patients. So the EGFR mutation rate in patients in China is about 40% versus about 10% in the United States. So you add that higher prevalence of mutation with the larger starting point of more patients in China and generally, it ends up being that there are about 10 times the number of addressable patients in China with the L858R EGFR mutation that our drug is targeted against. So we wanted to find a path to not only help those patients, but access those because we believe that could accelerate our clinical trial enrollment a faster proof of concept and as quick as possible access to the data to see if the molecule is working the way that we described it. I think the risk sharing one is common to any sort of regional out-license. One of the things that was really great as we navigated this process with MSQ is we discovered some opportunities to even lean in a little more on that. So when we made the decision to go to China, of course, the other option is to manage and pay for a CRO with capabilities in China. We felt like it would be more productive to have a partner that has shared and aligned incentives in all the ways where we could share risk and reward. And so when we kicked off the process, we were looking for a partner and ultimately found a great one with Betta, where they have excellent operational capabilities and proven capabilities in both the development and commercialization in non-small cell lung cancer, including 2 EGFR inhibitors. And so one of the discoveries that we made along the way was with Betta, they were interested and wanted to do more than initially I think we had planned on. And so where we've landed is we plan to initiate our clinical trials outside of China after Betta has completed dose escalation in China. And so we believe that's the fastest way to get through escalation, to get the data that can then allow us to jump into a multi-regional clinical trial, which will accelerate timelines and data access. And so C4 retains all rights to CFT8919 outside of Greater China. I think on the second question you asked around advice for Western companies seeking China partners, I think a couple of silly ones and some important ones, right? I think you need to be prepared for late night and early morning calls because a 12-hour time change with the US, at least in the Eastern time, makes for some both of those on both sides. Certainly, we found it productive to utilize folks who have cross-border expertise. So MSQ and our outside counsel both helped us navigate some cultural nuances and understand what it is. I think this goes for any deal, but I think in particular, one thing that was useful for us is we needed to understand what our goals and our strategy for seeking a partner in China, because there were a lot of organizations in China. And so I think it was useful for us to have a strategy and be able to compare the organizations that were interested in partnering with us against what our strategy and what our goals were to make sure that there was alignment there. I think one of the things that also is different is the Chinese landscape of companies is changing. I think, in 10 years ago, it was companies that were focused exclusively in China. I think there are a number now that are Chinese companies who want to have, of course, a Chinese presence, but want to become global, whereas there's also still some companies that want to stay kind of regional in China. And so depending on the nature of the agreement and the deal, those sorts of partners will be important. There's some differences, of course, with regulatory, with the cross-border support. I think one of the key learnings there is some of the health agencies have different criteria at different stages, which requires a little bit of navigation. I think the third question you asked was about some of the risks and benefit of the multi-regional clinical trial. I think one of the things that we saw and learned that it needs to be multi-regional. It cannot, a registrational study is unlikely to be successful if it just includes 100% of patients in China. We saw that with some other activities while we were going through this process. But there are a number of medicines that have been approved in EGFR and non-small cell and others where greater than 50% of the clinical trial patients that only go into the registrational study were contributed by in patients in China. And so these multi-regional clinical trials, of course, demand greater coordination across time zones and languages. But we believe that especially in EGFR, and I'm sure there are other cases where overall enrollment efficiency can be accelerated, and you can achieve the requirements for all of the health authorities with care and coordination. Certainly, there's been some improvement in harmonization among the health authorities where that is there. I think one of the other challenges that is important and is a nice puzzle to solve is with any sort of multi-regional clinical trial, the partnership model needs to align incentives and cost splitting. And so that was certainly a fun puzzle that Ginger and I were able to solve along with our beta colleagues that align those incentives for both parties. So maybe I'll stop there and follow up with any questions that are later.
Okay, thank you, Scott. Yeah, sounds like now you know China better than me after the collaboration with Beta Pharmaceuticals. And also I echo Scott. You just mentioned the culture thing. I believe it's very important. During my collaboration with C4, that's one thing I really appreciate, because like a C4 colleague always asked me, what is the culture in China, even for our like a celebration ceremony, and you guys say, what is the custom, right, in China for such kind of a celebrations? I think it's really important for each party, to understand the other's culture, to respect and appreciate that. Yeah, thanks a lot for sharing, Scott. So I just noticed Ro, can you hear us?
Yes,
Hello?
I can hear you.
Yeah. So we already have discussed a few questions. You just joined us. Maybe you can introduce to the audience a little bit about yourself and Kinto.
Yes, I'm sorry, I'm a little late. I got distracted tonight. I'm from Kinto Pharmaceuticals. It's a drug discovery company in China, established in 2009, and it's been 14, 15 years. We have been doing the PROTAC study for quite a while, because our company started with concentration on androgen receptor antagonist. As you know, the front runner in the PROTAC field in terms of clinical research is Avinas, and Avinas is doing androgen receptor PROTAC. So since we already have androgen receptor antagonist, we have the a recruiter for the protein itself, and so we decided to start doing PROTAC from that time. And we have done a lot of studies, but we kind of met the same problem as every other company has met. We have had some problem with the PK profile of our oral androgen receptor PROTACs. So since we've been doing a topical compound for alopecia, so basically we are already developing androgen receptor antagonist for topical use. We decided to take the advantage of this PROTAC, the big molecule that cannot be absorbed orally, and use it as a topical agent. So we decided to follow up with our topical androgen receptor antagonist as a topical androgen PROTAC, androgen receptor PROTAC, to develop it, to bypass the oral absorption problem of it. And since we really don't want this compound to get in the system to generate the some side effect related to as androgen receptor antagonist, so we actually taking advantage of this core property as our advantage here. So we developed a topical PROTAC. Right now is in phase 2 clinical trial in China. So this is, I guess, my brief introduction of our compound and what we are doing in terms in the field of the PROTAC itself.
Thank you, Ro. Yeah, we will circle back to you to have more detailed discussion. And now I wanted to shift our conversation a little bit because we have Tom, the CEO of Capital here, who actually is a very hardcore scientist himself. I spoke to Tom a couple of weeks ago. He has really good insights in this field. So I don't want to keep that knowledge to myself. So I was like, I have to invite Tom to share it with a broader audience. So to those who are not familiar with Capital, so Capital, as Tom just mentioned, is one of the very few companies working with both molecular glue and also bifunctional degraders. So both technologies are very exciting, right? And will bring a lot of new opportunities to the treatment paradigm. But in the same time, each has its own limitation and challenges. So Tom, I would like to take this opportunity to ask you to share, in your view, what are the pros and the cons of each technology, and also you can discuss how Capital intends to maintain its competitive advantage in this rapidly evolving space.
Yeah, thanks, Ginger. I mean, I think it's interesting that on this panel we actually have quite a few experts on both glues and PROTACs, but it's certainly an area which is reborn, because obviously the initial excitement, I think, was heavily in the beginnings of the new wave of TPD, if you like, after people figured out how Revlimid worked. The big attraction initially was PROTACs, but I think glues have gone through a big rebirth recently, and people are really understanding those much better now. But I would say that from our side, we have two bags, if you like, relevant to bifunctional degraders and to molecular glues. One bag is our pipeline, which is the kind of cereblon-based approach. And I think there, we've really tried to make the best of the, as you said, the advantages of each modality. And I think our pipeline actually is a good way of exemplifying it. So we have two glue programs, one of which is an IND-enabling study for hepatocellular carcinoma. And we have a second one in autoimmunity, which is very selective for NEX7. And both of those are glues. Both of those have great oral bioavailability. And in fact, we've even got series now with excellent brain penetration after oral administration. So to a point made earlier, we are thinking seriously of looking at neuroinflammation as a potential indication. And actually, it's interesting that recently we entered into a strategic collaboration with Ono Pharmaceutical, which is actually specifically on degrading a novel target involved in neurodegeneration. So this is a program we've been working on for about 8 or 9 months now, which is going very well. So we are very interested in things like CNS, particularly with glues, but also we think it's potentially feasible also, although much more difficult, with PROTACs, which is one of their problems, is that whilst glues tend to be a bit restricted in the kind of targets that you can address, and they're quite difficult to find, new glues. But on the other hand, PROTACs are much more modular. It's a bit like Lego. You take a ligase ligand and a target ligand and put them together, and hey presto, at least that's what people thought. But actually, the reality is they're very hard to optimize and get to good drugs. So that's one of the downsides. And in fact, our most advanced bifunctional, or PROTAC as our Venus call them, is a first-in-class, we believe, a first-in-class MCL1 degrader, which is actually took us a long time to optimize. So in contrast to our glues, where we could move forward very quickly to get to really good compounds, with our MCL1 PROTAC, it took a while to get a good compounds. But now we've got too many good compounds. And in fact, we're delaying a little bit our candidate selection because we have an embarrassment of choice. We've actually got some, which is hard to believe, but we've actually got some PROTACs here degrading MCL1, which have a DC50 of 100 picomolar, which for us is very potent and is rather unusual, to the extent that we're even thinking of applying them to ADCs as an ADC payload. So I think I would say that our first batch of if you like, of molecular glues and PROTACs, we're focused on making better drugs, really. We're focused on chemical stability, which was an issue with some glues. We're focused on the druggable properties, which can be an issue with PROTACs. We've looked at brain penetration. So we've really tried to make better drugs and particularly focus on this aspect of chemical stability as well as cell activity. So our MCL1 PROTAC and our NEX7 degrader for autoimmunity are both exquisitely selective compared to other glues and PROTACs, and we're very proud of that. But we're also looking ahead, again, thinking of glues and PROTACs. And this is where our new ligases come in, because we've actually synthesized about 100 and purified about 100 new ligases, the proteins. And then we've been working aggressively on 10 of those. And recently we made some big achievements in terms of, in particular with the Kelch family of ligases, because they've actually now got a production platform for those different members of the Kelch family. And we've got some very useful ligands now against some of those members. And only about a month or two ago, we actually successfully degraded using one of the Kelch ligases, degraded a target, so demonstrated a PROTAC ability. And some of the other ligases we're working on as well potentially are maybe even gluable because of the plasticity of the surface. And this brings me to the last point I'll make on this subject is that one of the key reasons we've really been successful in optimizing our glues and PROTACs is our huge focus on structural biology, on crystallography, on modeling, and all of those aspects. We have over 30 people who just do internally, who just do structural biology and crystallography. And as I said at the beginning, we've solved about 400 crystal structures down to around about 2 angstroms. And what we do is we use that iteratively to optimize. So we crystallize the ligase or crystallize the target, or even sometimes the ternary complex with our molecule fixed. And we use those structures to optimize, to design better molecules, and then iteratively to do new structural work. So it really is very labor intensive, but in fact, we find it very successful. And I think we are, as I said, we're very comfortable with both glues and PROTACs, and we're keen in our pipeline, which is cell cereblon based. And our next step now is to apply the learnings from cereblon to those new ligases, which we have got production platforms for and screening technologies for. So that's really the next step for us, is to take molecular glues and PROTACs to the next area, which is new ligases. And I guess finally, we made a recent breakthrough in that we actually got some new ligands for cereblon, which are covalent, which is relatively new for us. But this one is really interesting because it's a covalent ligand for cereblon, which actually is unique. As far as we're aware, it utilizes a residue on cereblon that's never been used before for covalent linking. It's traditionally not a covalent linker capable. So we're very excited about that, and it gives us some very nice and unique conformations of cereblon. So this is another whole new area that we're launching into in relation to even cereblon glues. So I guess that, that's what I wanted to say on that. But as I say, I think I'm surrounded by people here who know about both glues and PROTACs.
Thank you, Tom. Yeah, for this quite educational insights, and it's exciting to hear a lot of progress going on in the chapter. So today we also have quite a few representatives from China-based companies. As we know, actually, biotech companies in China are catching up quickly with new technology, right? Starting with CAR-T, and then ADC, and then now TBD. So with that being said, I believe it's critical for the audience also to hear from Chinese representatives about what is happening in China, to really hear their voice. I will start with Xiaobao, so the CEO of Glutex, which was founded in 2021. So Xiaobao actually led the firm and managed to navigate quite a challenging economic macro environment and secure the R&D in China and the US in a little bit over 2 years. So Xiaobao, can you share your journey, how you are able to attract the investors in this very tough market, and do you have any plans for partnership in the near future?
Okay, thanks. Okay, because GloTech Therapeutic is the youngest biotech, just built up 2 years. So I introduced our pipeline to prove our efficiency and how to abstract the investors now. Okay, now GloTech Therapeutics have five pipelines, 2 molecular glue pipeline. The first glue pipeline GT919 has shown great pharmacodynamics, pharmacodynamics and safety profile in pre-clinical study, and it has been approved to enter into clinical trial both in China and the US. We have just started recruiting patients in China and would love to have partner overseas to advance in the US. Besides GT919, we have also glue GT929 in NHL, which IND application have been accepted by NMPA now. We have also found novel targeted glue compound to validate it. Now I introduce our PROTAC pipeline. A first-in-class PROTAC GT838 that has just started a preclinical study. Hopefully, would be submitted to apply for pre-IND meeting later this year. Another GT868 is a best-in-class targeted PROTAC. And we have seen good selectivity in particular mutation cell lines. We have also one targeted PROTAC that is pointed by Chinese farmer, and we are going ahead to develop it jointly. Again, it's welcome if any potential partner could reach out and have a deep discussion with us regarding any cooperation intention. That's all. Thank you.
Thank you, Xiaobao. Yeah, for our audience, if you are interested in collaboration with Xiaobao, just feel free to reach out to him. So moving on to Leo, I noticed your company was also founded in 2021, right? And so far, you have raised $90 million. What is more impressive is that actually, all these investments are from quite big names. Qiming, LAV, Here House, and the Kai Tai, right? So one interesting thing I noticed that you have, as you just said, you have operations both in the United States and in China, which we know require substantial coordination, planning, resources, because you have to deal with different time zone regulatory officers, business modalities. So can you help us understand, the business of the benefit of this model and why you see this is a good option for Glue Bio? And also, do you have any comments on the advancements of a TBD in China versus the United States?
Sure. Thank you, Ginger. Yeah, I think the rationale for this kind of model is because the reality of the biotech ecosystem in US and China. For China, I think we have a very powerful and well-established ecosystem, especially in Shanghai. There are many CROs around us for chemistry, CMC, and enabling. They are very efficient. And so I think we believe if we could leverage the cell resource in China and also the cutting edge biology in US, especially in California, so we could be able to really speed up the research process and improve the efficiency. So that's the way we do the research. And like I mentioned at the very beginning, we recruit lots of well-trained and talented medicinal chemists and also computational chemists in Shanghai. But for biology part, I think we have a very strong team with 6 to 8 years experience in Merck, especially just like other biotech like Neomorph, which is also Glue biotech in California as well. So we kind of leverage all these strengths from both Shanghai and California. So that's the way we can move our asset very efficiently and very quickly. Just for one example, I mean, for our first and second asset in IND, for the first one, we already get the IND approval from FDA. Second one just filed the IND. All those 3 assets, we took only 7 to 8 months from DCA nomination, drug candidate nomination to IND filing. It's very efficient. That's the way, I mean, the global way, right? The second question you are asking is the enhancement of the TBD in China and in China. I think like you mentioned, there are a lot of progress for biotech in China or biopharma in China in TBD area. I just will give you one number, right? At this moment, there might be, I think, all the TBD protein in clinical trial or in the world, there might be like 30 drug candidates. And the China team or Chinese biotech or biopharma contribute 11. So that's 11 of 30. And 1/3 of the protein or bifunctional degrader in clinical trial come from China. But regarding to the glue, I think the bar is much higher. Merck glue is not that easy. Like Tom shared or Xiaobao shared. It's not a very straightforward technology. You have to have very strong bio team to figure out what target might be the ideal target to be degraded. And you have to also set up the screening platform to find out those targets and really understand the MOA and really understand the target product profile for Merck glue are the key for success. If you find some compound not selective enough, if some new substrate, we call new substrate or off targets, which was degraded and what degraded will cause a lot of tox. You don't even know. That would be a trouble for drawing clinical trial for Merck glue. So for our team, we have a very strong biology background, and several members actually origin from Celgene. They have moved several drugs in clinical trial. So that's some kind of know-how we have from team. But for chemistry part, I have been working for TBD for 7-8 years. Before I founded the company, I worked at J&J. So I built up the platform and I moved their program, first in class program program to in vivo study or POC study. So I think we are very confident this we can do this much better than any company in China, especially in glue and in glue field. Yeah, that's what I want to share. Anyway, just long story short, I think the protein is going pretty well in China and lots of good assets in China. So if anyone want licensing or collaborate with biotech or biopharma in China, there should be some very valuable assets. But for glue, I think it still needs some time to be more matured, right? For biotech, not only in China but also in US and in EU. The bar is much higher. I mean, it's not like protein, so everyone can do protein in the next 5 years.
Yeah, thank you. Yeah, I think you just demonstrated a perfect example for China's speed. Okay, so because we are actually hitting the time mark, I don't know any of you have to jump for another call, but I mean, it's not easy to gather all of you together. So I would really love to take a few minutes to hear about your crystal ball prediction for the next breakthrough in this field. I mean, because over the time limit, maybe we, each of you just, spend a few sentences to answer this question. Michael, do you want to start first?
Sure. So a lot of the things that the panel's already talked about, and going beyond cereblon and unlocking additional E3 ligases to widen the potential target space would be of high interest, right? And then going beyond the core therapeutic areas of oncology and then breaking into immunology and neuro, that's technologies that would allow us to develop molecules that would have brain penetration and predict in a predictable way. So, essentially novel modalities that go beyond our existing, PROTACs, including glues and autophagy-based approaches. So I think these are some of the directions, I think, for this space that we should expect to see in the future.
Yeah.
And Ginger, I do have to drop off, but thank you very much for
the
invitation.
Yeah, we'll stay in touch. Tom, you have anything to add? Oh, you're on mute.
Yes, it just follows on from what Michael said. I mean, we think that the future is in two ways. One is this whole potential opening up of the new ligase area. It's not going to be easy because there's a lot of ligases, and then you have to make the connection to the targets. But we started actually working on new ligases in about 2018. So we've got, as I said, about 100 of them. So we're persevering, and we now made this breakthrough with this family, and we've now have shown demonstrated PROTAC. So that's very exciting for us. But we think there's also some good, and by the way, so some of those ligases, for example, that we are working on are overexpressed in cancer. There's a few other particular aspects of them which could be quite interesting. I think it's probably not going to happen that we'll find an exquisitely selective ligase. I think they probably don't exist. But I think we can look at preferential. And finally, I think novel approaches with cereblon are still going to be worth following, and in this whole area of potentially new covalent ways of approaching cereblon. And also what we've found is that we can really narrow down the specificity of glues and get rid of those substrates that you don't want to degrade. So all of those kind of things are really important, I think, to keep going. Don't forget cereblon. I think new ligases are going to be a big future.
Yeah, that's quite an interesting selective ligase. Yeah, that's something we can explore. Ro, Shiro, do you want to add anything?
Yeah, I like to say, I mean, right now the field is really hot. I want to say that there'll be some obstacles in the way towards the actual drug. I mean, we got a long way to go. Right now, everybody's concentrating on VHL and CRBN. They all have their own problems. I think Tom is right. The public future, at least to open up the field a little bit, is probably going to go for some new ligases. I found out that all the glues that is concentrating on the CRBN, cerebral, and I mean the molecule. If you look at all the molecules from different groups that become more and more similar. And because CRBN, that's as the previous speaker said, that they actually, I mean, the glues that very peculiar, that sometimes a very small change can have a lot of unpredictable changes that in terms of the properties, especially in vivo properties. So I felt that we still got a very long way to go. I mean, I'm very excited about the new field, but we have to be patient. That this is not saying that we already got a new drug on the way. Once we have a first new PROTAC on the market, then we can celebrate.
Okay, let's see. Let's be patient. Xiaobao, do you have anything for big prediction?
Okay. Although GluTech Therapeutics have two pipelines, that is, the one is glue, the other one is PROTAC, but we are continuously pay attention to new technologies in TPD field. For example, we have drawn attention to a newly designed degrader that can directly recruit proteasome to degrade the new substrate. It has the potential to overcome the E3 ligase acquired drug resistance. which might be a niche market after first generation TPD drug enter into the market. I think biotech should lay good foundation to solidify the R&D platform and differentiation strategy. They should also actively seek for collaborations from big pharmas, use unique clinical and commercial experience to accelerate the pipeline into market. Now, I believe the degrader looks like the good rush is on.
Okay, I share the same optimistic with you. Leo?
I agree with all these comments from our speakers. For me, I think protein is going pretty well. I guess hopefully coming in phase 2 and phase 3 clinical trial will be improved in the next 2 or 3 years. That will be a breakthrough and breakthrough news for TPD field. Regarding to MSQ, I think this kind of a modality is still and is in its infancy, right? Because we know there are still more images on the market or in phase 2 or phase 3 clinical trial. However, I think for other new substrate or new targets, for MSQ is still at a very early stage. We need more time to really validate those targets and find the right compound for those targets. That takes some time, right? But regarding to product, hopefully some like IR4 from Chimera or ERAR from Avidas, they can move forward to phase 3 and get approved by FDA. That'd be very cool, I guess. But overall, I think I'm very confident that the type protein integration will be the most promising and productive technology, especially for small molecule drug discovery. So yeah, that's what I want to share. Thank you.
Yeah, it looks like the world is trying to see how Venus is going to when they can cross the finish line. I'm going to talk to Randy about this. He will be on our other panel. No pressure for him. So lastly, Scott, do you want to wrap up today's conversation?
Sure. So I think the last kind of the crystal ball one that I'm thinking about and that we're thinking about is similar to something that Tom brought up earlier. While we've spent a lot of effort utilizing advanced medicinal chemistry techniques to make our molecules orally bioavailable, we all discover a lot of molecules that may not achieve that. And so I think one near-term frontier is ADC degrader combination. Certainly, there's been a lot of talk and buzz around ADC and success with a number of publications showing it. I think one thing as we've been having conversations and navigating some of that is the collaboration and the partnering piece is where there may need to be some innovation because it needs to align the innovation coming from both parts. The degrader that's designed for as an ADC cargo as well as monoclonal antibody. And so I think that's a near-term frontier where it can unlock some of these great degraders that have been discovered that may not have legs as orally bioavailable molecules. So
that's interesting. Yeah, I mean, now we have so many ADC companies and a lot of protein degradation. I mean, it would be a fantastic combo, right? Okay. All right. Thank you, everyone, for contributing to today's panel. And I wish you guys have a good day, good night, and a good afternoon. And I will see you guys soon. Thank you very much.
Okay. Thank you.
Thank you.
Everyone.
Thank you. Bye-bye.
Bye.